So now we embark on our final unit. It's all about reproduction and development. This chapter will be about the male reproductive system predominantly, and the next, the female reproductive system, and then we'll look at embryonic development in our final chapter. So the essence of sexual reproduction is that it's biparental. So offspring receive genes from two different parents. Offspring are not genetically identical to either parent, but they do contain genes from both parents. Eventually, we die, and our genes will live on in a different container in our offspring. The gametes are produced by each parent. These are the sperm and the egg. When the sperm meets the egg, we form a zygote, which has a combination of both parents' genes. With the two sexes, it's important that one gamete has motility. For us, that's the sperm and is found in the male. The parent with the Y chromosome is considered to be male, genetically. And the parent with no Y chromosome is considered to be the female, genetically. Of course, there's some variations on these dependent on whether receptors are present for testosterone and so on and so forth. We'll take a look at a couple of those. The female gamete is the egg. In mammals, the female is the parent that provides a sheltered and internal environment and prenatal nutrition for the embryo. Both sexes have copulatory organs. The male's is the penis, and it's designed to introduce gametes into the female reproductive tract. The female copulatory organ is the vagina, and it's designed for receiving the sperm so that the egg and sperm can unite. So in short, the male reproductive system is designed all around producing sperm and introducing it into the female body. And the female reproductive system is all about producing eggs, receiving sperm, providing a space for the union of the gametes, and then a space for development of the fetus and the equipment to nourish the offspring during its growth. The reproductive system is consisting of primary and secondary sex organs. The primary sex organs are the gonads. They produce the gametes, so the testes and the ovaries. Secondary sex organs are anything other than the primary sex organs that are necessary for reproduction. So in the male, we'll see there's a whole system of ducts and glands that help the penis deliver sperm cells. In the female, we'll see that there's the vagina that receives the sperm, and we'll see the uterus, which is for the developing fetus, a whole system of tubes that allow the egg to move to the uterus. The external genitalia are located in the region of the perineum. That's inside this diamond shape right here. The corners of the perineum are at the ischial tuberosities or sits bones, the tip of the coccyx, and the pubic symphysis. In this region is the perineum. Most of the external genitalia are visible. However, in the female system, there are some accessory glands that we cannot visualize. The internal genitalia are located mainly inside the pelvic cavity. Except for the testes and some of the associated ducts in the scrotum, they have become specialized to descend out of the body cavity to accommodate specific temperature requirements for sperm development. The secondary sex characteristics are features that are going to distinguish the sexes and play a role in mate attraction. Most of them develop at puberty. In both sexes, that would be the axillary hair and associated scent glands, as well as the pitch of the voice. In the male, at puberty, we see facial hair development, coarse and visible hair on the torso and limbs, and a relatively muscular physique, whereas the female will see a distribution of body fat, breast enlargement, and relatively hairless appearance of the skin. Now, sometimes you can have a genetically male individual that expresses a female appearance. This is the case of androgen insensitivity syndrome. Sometimes a girl will show all the usual changes of puberty but will never have menstruation. And so if the age of 16 passes and menstruation hasn't appeared, then a simple test of karyotype, that is, what is the chromosomal composition, can reveal whether indeed it's a case of androgen insensitivity syndrome. These individuals all have the karyotype, or the genetic sex of male, XY. And they produce testes and normal male levels of testosterone. However, none of the targets have receptors for the testosterone, so it's as if testosterone isn't even there. We'll see that female is the default sex. That is, if there's no testosterone present or no testosterone detected, we'll see development of female external genitalia. However, there's no uterus. It's a blind vagina, shorter than usual. And if there's no uterus, then of course there's no menstruation. So that's a nice introduction into looking at chromosomal sex determination. All our cells have 23 pairs of chromosomes. 22 of them are autosomes, and one pair of them is sex chromosomes, XY in males and XX in females. Males produce about half Y-carrying sperm and half X-carrying sperm. And reproductively viable females only generate X chromosomes. Thus, the male determines the sex of the child. So now let's look at how sex differentiation happens. It all has to do with some prenatal hormones. So initially, a fetus is sexually undifferentiated. This is the undifferentiated stage here. Gonads will begin to develop at about 5 or 6 weeks from gonadal ridges. Here, they're neither testes nor ovaries. There are 2 sets of ducts adjacent to each gonadal ridge. Here, we've got the paramesonephric duct and the mesonephric ducts. The mesonephric ducts will develop into the male reproductive system, and the paramesonephric ducts will degenerate. However, if the fetus is to be female, the paramesonephric ducts, or Mullerian ducts, will develop into a female reproductive tract, and the mesonephric ducts will degenerate. So this is all dependent on the presence of the SRY gene, and the SRY is a very small region, the sex-determining region of the Y chromosome. And so naturally, it should be found in males. It codes for a protein called testes determining factor that initiates the development of testes from these gonadal ridges. We should begin to secrete testosterone from these early testes around 8 or 9 weeks. The presence of testosterone will stimulate the mesonephric ducts to develop into the male anatomy. At the same time, the testes are going to secrete Mullerian inhibiting factor. And Mullerian inhibiting factor will cause degeneration of these purple paramesonephric or Mullerian ducts. So now we're left with testes and all the ducts that come from the mesonephric ducts that support transport of sperm. Estrogen levels are always really high in pregnancy. So if estrogen was the hormone that directed female development, all fetuses would be feminized. Female development is going to occur in the absence of androgen hormones. So in the absence of SRY gene, we will see that the default is that ovaries develop, and because there's no Mullerian inhibiting factor, the Mullerian ducts or paramesonephric ducts will become the oviducts and uterus. And if there's no testosterone to support them, the mesonephric ducts will degenerate. And thus we have development of the female internal genitalia. Development of external genitalia occurs in a very similar fashion in both sexes. There's 3 root materials: the genital tubercle, which will either become the head of the penis, the glans, or the clitoris. A pair of urogenital folds that you see here in pink that will either become the urethra of the male and forms the penis, or it will form the labia minora. The urogenital fold is composed of erectile tissue. And then there's a pair of labioscrotal folds that can either become the scrotum or the labia majora. So all of this again depends on the presence of testosterone. If testosterone is present, then we'll see development of male external genitalia, and if it's not, we see the development of female external genitalia. So it's surprising, considering all of these different processes, how often we actually do come up with genetic XY male and genetic XX females that reflect the actual genotype. Some babies are actually born with undifferentiated genitalia, and so the scrotum hasn't quite closed, so it looks like labia, not really sure about what the sex of the child is. So here a karyotype will be necessary, and then they can decide what way to fix the external genitalia. However, usually by week 12, either male or female genitalia have been distinctly formed. These structures that develop from the same embryonic tissue are called homologous structures. So the penis is homologous to the clitoris, and the scrotum is homologous to the labia majora. And the labia minora are homologous to the corpus spongiosum of the penis. So both male and female gonads initially develop high up in the abdominal cavity, very near the kidneys, and they migrate into the pelvic cavity or the scrotum. The gabernaculum is a connective tissue cord that extends from the gonad to the floor of the pelvic cavity. In the male, it passes between the internal and external abdominal oblique muscles and out into the scrotal swelling that you can see here. At about 8 months, the vaginal process will develop. It's a fold in the peritoneum that extends into the scrotum through the inguinal canal. The inguinal canal is a pathway of fairly low resistance through the groin, and it's created by the gabernaculum, this string of connective tissue, and the vaginal process. It's the most common site of herniation in boys, an inguinal hernia, where the contents of the abdominal cavity can get pushed down through the inguinal canal. The inguinal canal is where also the major blood vessels will exit the body cavity and make their way down the legs. So testes descent begins as early as 6 weeks. During the 7th month, the testes abruptly pass through the inguinal canal into the scrotum, guided by the gabernaculum. So literally, the gabernaculum, this ligament, is pulling the testes down towards the scrotum. The testes are moving down through the inguinal canal with the testicular arteries and veins, the lymphatic vessels, the nerves, all the ducts that supply them, and extensions of internal abdominal oblique muscles. Cryptorchidism is a condition where boys are born with undescended testes. It occurs about 3% of the time. Most cases, the testes can descend during the first year of infancy. However, if they don't, an injection of testosterone can help them descend, or a simple surgery to literally draw the testes down into the scrotum. And it's really important to fix cryptorchidism because it can lead to sterility and testicular cancer simply because the testes are trapped inside the abdominal cavity and the temperature is simply too warm for them. Ovaries also descend, but to a much lesser extent. The gabernaculum will pull them down and they'll lodge on the inferior brim of the lesser pelvis. The gabernaculum becomes a pair of ligaments that support the ovaries and the uterus. So now take a moment to work on our first diagramming component for this chapter. We're going to diagram sexual differentiation. Things to consider. We go back to this figure here. Sexually undifferentiated stage, and what causes the change from undifferentiated to differentiated include things like the mesonephric ducts and paramesonephric or Mullerian ducts, and definitely address the presence of SRY. If SRY is not present, then we have no testes determining factor, and thus no testes, and thus no testosterone. However, if SRY is present, then we do see testes determining factor, and we do see Mullerian inhibiting factor. So make sure you have many of these terms in there and see what you can do to pull together a diagram of sexual differentiation of both internal and external genitalia. And I'll see you in the next section where we examine male reproductive anatomy.